DNA is located inside the cells of all living organisms, as we already know. The nucleus, the cell's information center, contains the majority of DNA in animals, plants, and fungus. Using common home items like washing up liquid, salt, and alcohol, we can extract DNA from fruit in this activity. We'll be utilizing essentially the same chemicals and procedures that are utilized in the lab to extract DNA.
Good day everyone. Today we are going to extract DNA from fruits. So, pay close attention if you are interested in doing this experiment in your home. I hope you enjoy.
First, we need to prepare the materials needed for this activity. We are going to be using a ripe banana fruit for this one. However, if a banana is not available, kiwi, raspberries, strawberries, or even tomatoes, those will also do.
We need to prepare a banana fruit. 3 cups, which are preferable transparent, a knife, 1 tablespoon and 1 teaspoon, a sifter or strainer, a funnel or tissue if it's not available, a narrow transparent container, dishwashing liquid, water, and lastly, an alcohol which should be at least 70% isopropyl alcohol.
After preparing all the needed materials, you can now proceed. First thing that you need to do is to peel the fruit. In this case, peel the banana using your hands or any method that makes you comfortable. After peeling it, cut it in half and then you have to chop it finely.
Next thing to do is to mash the banana using a spoon in a container until it turns into a puree, or if it's available, you can use pestle and mortar.
Next thing we need to do is to prepare the lysis solution. Mix in a container 1 teaspoon of liquid salt and 5 tablespoons of water.
Then, pour the lysis solution into the kiwi puree.
Since the DNA is trapped in the cells of the banana, we can break it loose through the dishwashing soap by disassembling the membranes that are made of fats.
Make sure to mix it very well for at least 5 to 6 minutes so that as much DNA as possible is released from the cell.
Filter the mix using a French strainer or sifter. If you have no sifter, you can use a filter method using a tissue. The DNA will be dissolved in the water that pours through, which is the reason why we added the water.
After a while, you should end up with a crystalline or semi-crystalline liquid. If you use the tissue method or proper filter, a clearer solution will appear.
Transfer the filtered liquid into the transparent container. This liquid contains the DNA of the banana dissolved in it.
Next thing we need to do is to precipitate the DNA, which means take it out of the solution through putting a decent amount of alcohol. It's important to use cold alcohol because it allows a larger amount of DNA to be extracted. If the alcohol is too warm, it may cause the DNA to denature or break down. The water that is now dissolving the DNA is going to instead go with the alcohol, so the DNA has no option but to precipitate.
As a result, a white slime should appear forming in the middle of the alcohol layer, that is the banana DNA.
By any means, it is not pure DNA. It has RNA, proteins, and other bits and pieces that got dissolved in the water and then precipitated in the alcohol, but it is mostly DNA.
You can try to fish out some of the DNA using a stick or a cotton bud. Since DNA is a long molecule, the DNA of one cells get entangled with the DNA of other cells, which makes it very easy to pull them out.
We deliberately use the minimum possible number of reagents, but you can add a few more for further accuracy. For example, you could add a pinch of salt right before adding the alcohol to aid the precipitation of DNA. But as you can see here, it precipitates fine without. Similarly, you can add a few drops of pineapple juice to the filtered liquid or to the lysis solution to eliminate some proteins. This is because pineapple has bromelain, an enzyme that breaks up proteins, so this would give us purer DNA.
However, it would not look very different, so we decided to leave it out in this case in order to have a simpler protocol and just mention that the DNA is not pure. Also, it would have the obvious problem of having a second fruit into the mix with its own DNA. Alternatively, you could use contact lens washing liquid, which also has proteases, but this introduces a black box reagent that we would rather avoid.